All right, let's get started. It looks like the participant numbers starting to die down, so we'll get things going. My name's Pat Shepard. Welcome to our Lunch and Learn series here at the Maine Center for Coastal Fisheries. I'm joined today by Simon Thurl, the senior scientist at Woods Hole Oceanographic Institute, as well as Robin Linier, one of the graduate students at the University of Maine. And we're going to be talking to you about codfish today. And our sampling program for groundfish in general. You'll see Tate Yoder as well, and he's going to field some of the questions at the end and run tech for us and make sure everything's running smoothly. We've got a lot to go through in an hour, so I'm going to bust through my slides so that we can get to the meat of this and what everybody's hopefully here to see, which is a live dissection of cod. We're going to do that in the shop downstairs, so definitely stick around to the end and check that out. So a little bit of background for those of you joining us from the Acadia Senior College. This is going to be a repetition of the slides that you guys saw on Wednesday. So think of it as a good song on the radio that you've been dying to listen to again. And we'll move on to Robin and Simon as quickly as we can. So when we talk about groundfish, a groundfish is not a particular species, but rather a suite of species of finfish and flounders that all sort of cohabitate on the bottom of the ocean and feed on the ocean floor or near the ocean floor. These fish are typically all caught together most of the time. And since they're caught together and they live together, they're also managed as what we call a stock complex. So there's about 15 different stocks of groundfish species that are all managed together in what we call the Northeast multi-species fishery. And those are things like cod and haddock and pollock, all of the flounders, so your halibut, dabs, gray sole, things like that, and also redfish. So when we talk about groundfish, we're talking about that whole suite of species that sort of cohabitate together. The groundfish fishery has actually collapsed multiple times over the course of history, and this chart shows the most recent collapse since about 1950. I was born sort of right in the middle of this in 1987. And so I was alive through the current collapse, although I didn't really appreciate what was going on around me. I was pretty young. But my first groundfish trip, I went on when I was about 7 years old with a local fisherman here in Stonington, the hometown that I'm from. And his name was Brent Oliver. And I was too little to be out on deck. I was with my brother on that trip, and both of us were sort of stuffed up in the wheelhouse, probably eating junk food all day. But I do remember one thing from that trip, and that was that we caught a monkfish in the gillnet. And if you've ever seen a monkfish, they're probably about the ugliest critter you've ever seen on the ocean floor. They've got these huge jaws with these massive teeth that are like inward turned. And he threw the monkfish up into the wheelhouse at me, and I thought it was going to eat me. So that was pretty fun. It was also the only thing I remember about that trip. Pretty traumatic experience for a 7-year-old, but here I am studying the things now. So the reasons for the collapse, I think are due to a few different reasons. Overfishing, we can certainly blame for part of it. There's a local fisherman here in town who was helping me build gill nets for the survey. And he recalls a story from when his kids were young. He was home for the first time for supper in weeks. I mean, these guys fished really hard for groundfish up through the 80s and 90s. And it had been weeks since he had been home to eat dinner with his family. So it was kind of a rare occurrence. And they were all sitting around the dinner table, and his son, who was about 6 at the time, got up from the dinner table because he was finished with his supper. And his father reminded him to clear his plate. And he looked at his father, and then he looked over at his mother, and he said, "Mom, do we have to listen to this man?" And it was sort of the first time that he had realized how much he had been sort of missing from the family, because he had been fishing so hard and so long. He'd spent just weeks and weeks out at sea. So overfishing and advancements in technology definitely have played a role in the demise of the fishery, at least here in Eastern Maine. But I think one of the bigger reasons that we can point to as a sort of a fundamental lack of understanding of their ecosystem, and in particular their prey species, which is what we're all here to talk about today. What did the cod eat for lunch? So back in the 90s, one of our founding directors, Ted Ames, recognized a link between codfish and alewives, especially as an important prey base for local stocks of cod that exist here in Eastern Maine. And after he recognized this link, it sort of was a turning point into understanding this fish's biology and its ecology and sort of how it exists in its ecosystem and the different factors that are at play when we talk about rebuilding. Alewives are a species of river herring, and they're considered anadromous, which means they spend most of their lives in the ocean, but they also spawn in the freshwater lakes and streams. And the adults will do this in the spring. They'll sort of travel into the freshwater lakes and streams in the spring. And then their young will travel back to the ocean in the fall. And that cycle happens about every 4 years. Well, it happens every year, but the adults, it takes a young alewife about 4 years to reach adulthood and reproductive maturity. So they return back to their natal places to spawn after about 4 years. There's a problem with that. Actually, there are several hundred problems with that. And that's in the form of dams that exist in virtually every river and every stream in Maine. There are literally hundreds of these things. And some of them have some sort of fish passage and some of them don't. Some of them have a little bit of help in the form of trapping and trucking. This is a graphic depiction of the number of dams that exist here in Eastern Maine, and it's just a massive number of them. So alewives trying to make it upstream to spawn are faced with a whole host of different impedances that disallow them to do that. This next slide is showing you sort of the different rivers that are accessible or inaccessible for alewives, and I've color coded them by green, yellow, and red. So green are the streams and rivers that are currently accessible to alewives. Yellow is accessible, but with some help. So there's either a trapping and trucking effort or a stalking effort, or sometimes literally people just volunteer to dip these things out of streams and then carry them physically up to the upstream portion of the dam and let them go. And then of course, red is completely inaccessible. And you can see a lot of that inaccessible habitat is on the Penobscot River system right there in the middle of the state of Maine. I'm going to steal this analogy from a board member of ours, a former board member of ours, Bob Stenick, who sort of relates this conundrum to the circulatory system in the body. And when you start clogging up different really important arteries in the body, you obviously get some health problems. And the dam situation in Maine is basically creating a heart attack for species of sea-run fish that are used to returning to their natal places to spawn. So how do we alleviate that? We do that by tearing down the dams that are here in Maine. The unused hydro dams usually are not used for anything anymore. A lot of them aren't used for anything anymore. So a lot of different organizations across the state are in the process of removing those dams. This is a picture from the Great Works Dam in Old Town. This is probably one of the biggest examples of a success story in the resurgence of alewives. Back in 2012, when the dam was removed, about 5 years later, 1.2 million alewives returned to that system to spawn, up from virtually zero about 5 years before that. And then last year, that number was about 2 million. So it's absolutely working. Now we have this interesting task of trying to figure out whether or not it's working, especially when we talk about the predators that exist in the marine system. So that's great, but how do we track the recovery of the system? Well, we have to figure out what's eating these alewives and whether or not our groundfish predators are feeding on them yet. We're seeing a massive resurgence of these young alewife populations in the state of Maine, and we want to try to track that recovery over time. So just a quick recap before I move on to the other presenters. Groundfish have been vacant here for about 20 plus years. We know that there's a historical link between alewives and cod, and we are hoping that that's still the case. We're hoping that by building these populations back up, that we see a predator resurgence back in the bays. We know that Eastern Maine has a distinct biology, and we know that it has distinct biological characteristics just because of the way the currents are set up, and the ecology that exists on the coastal shelf. So it makes it a unique place to study. And we know that alewife recovery is happening in the fresh water. So all of those different things point toward this sort of hypothesis that we have, that if we bring the alewives back, we'll bring the predators back as well. So how do we do that? Well, we set up a survey to be able to track that. And the survey has been going on for about 10 years now. And it's called the Eastern Gulf of Maine Sentinel Survey. And it uses longline and jig gear in the Eastern Gulf of Maine. And I'll let Robin take over from here and explain sort of the details of how that survey is set up, the different samples that we take, and then we'll move on to Simon. So Robin and I have, I've had the pleasure of working with Robin for what, 3 years now? 2 years. 2 seasons going on 3. And she comes to us from the University of Maine. The University of Maine provides an onboard observer and graduate student to this project every year. And Robin's going to walk us through her part of the work in what she does every day on the survey. So, Robin.
Hello, everyone. Thank you for having me. Okay, here we go. All right. So first, just to talk about the Sentinel Survey a little bit and kind of how it's set up. So these are the 2020 stations that we sampled last year. You'll see different colors, which basically just describe a different combination of the type of gear that we use. So Pat mentioned that we use both jigging and longline gear, but it also describes different depth strata, which, just to kind of describe what a stratified design is that we essentially break down kind of this range of depths into different categories, if you will, or strata 0 through 3. And what we do is we specifically choose a certain number of stations to sample within each strata, which does a couple of things. First of all, it makes sure that we are getting, this entire range of depths, and that we're not just looking too far inshore or too far offshore or right in the middle, because we want to be able to draw conclusions about this entire area. So it kind of helps us, make sure that we're covering all of the different depth ranges. And additionally, since we're sampling the same number of stations within strata year to year, it makes comparing to past years much easier. And so that's kind of why a lot of scientific surveys are set up in this way, because it helps try to reduce bias and keep things really consistent. So we've got longline stations, which are the blue ones that you see, which are split between stratas 1 through 3. We've got inshore jigging stations, which are the red ones, where we only jig and they're only in strata 0. And then offshore jigging, where we still are only jigging, but those are split between stratas 1 through 3 as well. And in addition to these random stations, we also have fishermen's choice stations, where essentially within this study area, fishermen have a limited number of stations in which they can just go and kind of choose the spots that they think are the best places to target some of our target species, kind of, outside of the bounds of this kind of random design. And that information is used in a little bit of a different way, but is still really important, especially for collecting the biological samples that I'll mention in a moment. So Sentinel Survey objectives, Pat mentioned, that this is a really great platform to ask some of these diet questions, but kind of overall objectives are a little bit more broad, and some of these kind of diet questions have come on the periphery. So to talk about kind of what the Sentinel Survey is trying to do overall is basically try to fill in some gaps where the state and federal surveys just can't sample. So as I'm sure you all know, there are lots of lobster traps in the Eastern Gulf of Maine, and there's a lot of really gnarly hard bottom, where if you try to pull a trawl or, a giant net through the water, it's going to get destroyed. So by using hook gear like we do, we're able to sample in some of those areas, which can be really important because things like cod prefer that like really hard rocky bottom. So, we could be missing information if we're only looking at the trawl surveys, since they just can't really get in those areas. And so there's kind of two major outputs. One of them is developing abundance indices, which is what you see over here on the upper left. And so we're not trying to actually guess like the actual abundance of these species, but what we do is, since we have that random stratified design and we can compare year to year, these abundance indices can be compared to each other so that we can try to see whether we're seeing increases or decreases, whether that's year to year or completely over time, to kind of just get a sense of whether we're rebuilding or whether things are getting worse. And we do that for all 4 species. I'm kind of focusing on Atlantic cod because that's the focus of my PhD and we're dissecting a cod today. But just to remind you guys, this survey also targets white hake, cusk, and halibut, which are all groundfish. And in addition to those abundance indices, we also want to know habitat information, where we're finding the majority of these species. So down here, I have a couple plots for cod, which essentially this one on the left is showing sediment type. So hard is going to refer to things like really rocky bottom or gravel. And that bar that's higher shows that there's a much greater chance of catching cod over that particular type of sediment. And we see it much lower for mixed or soft sediments. And we find that we catch the vast majority of cod in these kind of shallower areas, less than 50 meters, but that we do catch them between 100 and 150 as well. So getting into some of these kind of periphery studies, Simon's going to talk about this in just a moment, so I won't get into it too much, but we take tissue samples essentially from kind of just ahead of the dorsal fin on the fish's body, which is going to be used for stable isotope analysis, and we take that from all of our study species as well as some of our most common bycatch species, which are things like pollock, mackerel, and haddock. And some of the samples that I'll be showing you that I'm taking later today are morphometric photos, like the one that you see down here, which are used from scientists at GMRI. We take otoliths, which are little ear bones that tell us the age of a fish. We take stomach samples, which I'll get into some of those preliminary results in a moment. And then fin clips, which basically come from the tail, from the caudal fin, which tell us information about genetics. So we can extract DNA from those. And that has gone to scientists at UNH and DFO Canada. Again, I'll kind of get into these a little bit more later as I'm dissecting. So hopefully I can answer any lingering questions you guys may have about those samples. So part of my PhD is looking at cod stomachs. We are curious about what they're eating. There's been a changing ecosystem, right, with dams, with the overfishing of a lot of these groundfish species. And so we're really curious kind of what it is that cod in this eastern region are eating. So right now I have about 200 cod stomachs, and while we're on board and we take those, we put them in a 95% ethanol solution because it's really messy to try to analyze that on board. A lot of times I need things like microscopes to really figure out what's going on. So I've only actually analyzed about half of those at the moment. And I've attached essentially one of my data sheets here to kind of show you guys how I go about analyzing a stomach, which is writing down the prey type. So this first one was a lobster. Prey number, we always get a prey weight. Prey state is how digested it is. So F would be fresh, basically it isn't digested at all. P would be partial, and W is well digested. So you can see here that this particular fish had a lobster, a cunner, Cancer borealis, which is a Jonah crab, some seaweed, and what I could only define as crustacea, because that one was very well digested. And you don't want to try to guess what something is in a stomach in case you're wrong. So you just define it to, kind of as specific a level as you can, because that information is still really important. So looking at some of these preliminary results from those 93 fish, we see that crustaceans, which again are things like crabs and lobsters and shrimp, make up the majority, especially by weight, of the stomachs that I've analyzed, followed by fish, and other things like mollusks, worms, echinoderms, which are things like sea stars and sea urchins. But it's also really important to look at prey, not just by weight, because that's going to be kind of skewed towards the heavier things like crabs and like fish. And so down here, I've looked at the frequency of prey items found in stomachs. So how to read this is essentially in 52 of the 93 stomachs analyzed, I found at least one crab. So now we're kind of looking over the whole group of fish and seeing kind of where there are similarities in what they're eating. So crabs and shrimp are very common, followed by mollusks and fish and lobster. And so just to dive into that a little bit more, within crustaceans, we are mostly finding cancer crabs, which Jonah crabs are a part of that. And we're finding that basically cod of any size are eating these. And as far as lobster, it's usually the larger fish. As you can imagine, the larger a fish gets, the larger prey it can consume. Although we have found some fish that are eating lobster that are actually much smaller than we would guess. So in this case, as far as crabs and lobster, I mean, these are just very common things in the environment, that it's likely that this is just kind of what is the most available. And that's probably why they're the most frequent. However, we are still finding quite a few fish in the samples, even though most of the cod that we collect are what we would consider immature. That they're not reproductively, ready to spawn. Most of the time, they're not even, kind of growing their gonads. And there has been this idea that when cod move from being immature to mature, this is kind of when they start eating fish. But I've actually kind of found that it may be a little bit earlier than that. And so we found mostly Acadian redfish to be the most common. So I've got some pictures here of the cod and the fish that they were eating, the fish that we found in their stomachs. So this one that you see that I've defined as 59 centimeters and 5 pounds, she was a developing female. So she was developing eggs. And I don't even remember exactly how many there were, but I think there are like 15 little fish there. I mean, that cod was eating very well. And then the one underneath it, around the same size, this one was a male that was also developing its gonads, had a very good sized Acadian redfish in its stomach. So these are some really interesting kind of preliminary results. Obviously, I have a lot more to go through. Kind of drawing in from what Pat was saying, I have not personally found any evidence of alewives in these stomachs. And other researchers that are doing similar studies have not found that either. So it does kind of raise the question, if the alewives are coming back, why aren't we finding them in cod stomachs? And it could be a couple different reasons. It could just be that it hasn't been long enough, but just to kind of tie that together, that at least so far in my research and most of other researchers nearby, that we haven't necessarily defined that link, at least to cod, as being feeding on the hopefully returning alewives. I think that's all I have. So I will turn it over to Simon, and I think I can advance the slides. Yeah.
All right, thanks, Robin. That was great. Really cool to see. Yeah, so I'm going to talk about how we're using stable isotope analysis to understand more about this question. It's to us, it's a really interesting question, right? Because my sort of formative thoughts on this came from a lunch with Ted Ames in Stonington, actually, where we were eating Icelandic cod, as it turned out, on our fish sandwiches. But yeah, it's a really interesting question, and it really deserves an answer, because I think from my perspective, the ideas of removing dams from rivers is such a good idea, and the fact that we see these remarkable recoveries in alewife populations with the removals of dams suggests that if the dams calm out, the alewives will do better. And it doesn't matter whether you're in Maine or I gave a very similar talk down here on Cape Cod, and it's the same questions, right? If we can link that freshwater productivity that young of the year alewives bring back out of the rivers into coastal groundfish populations, then I think we just have another really strong motivation for continuing river restorations in the sense that, you know, if we do it, the fish will come. So anyway, this is a project that we're doing in collaboration with the Maine Center for Coastal Fisheries and with Carla in particular. So let's get into it. Next. So what are stable isotopes anyway? And so this is not going to get super chemistry-ish, but just really quickly, we're going to be talking specifically about carbon. So let's talk carbon, right? So carbon has different isotopes. In other words, we have the same number of protons in the nucleus of these carbon atoms, but we have different numbers of neutrons. And so we have either 6 protons, 6 neutrons, or 6 protons and 7 neutrons. And so just that extra neutron changes the mass of these carbon atoms enough that we can actually, we can measure the differences. And why that becomes really interesting is that just that slight change in mass of those 2 different atoms means that biological and chemical interactions in the body will preferentially use either the lighter isotope or the heavier isotope, depending on the chemical reaction. And that actually provides a really interesting way of tracing carbon from prey into predators as it travels up the food web. And so that's what we're going to use, but it totally relies on this very slight difference in mass between those 2 different kinds of carbon atoms. If we get over the next slide, it seems like it's jumped out of screen sharing. Robin, has that done that to you?
We lost connection for just a moment, so let me
okay.
Get back up. Are we ready for the next slide?
There we go. Yeah, let's go to the next slide. And so how do we use these differences in these isotope differences in ecology? And the answer in a sort of brief nugget is that you are basically what you eat, right? So the carbon that you're eating will be reflected in the tissues that you produce yourself. So as we go in a marine food web, for instance, from phytoplankton through copepods, through planktivores, through piscivores, we get regular and persistent changes in the isotopic composition of, say, muscle tissue in those different species. And so we use that predictable trophic fractionation, as we call it, to make inferences about the food webs that these fish are feeding in. And so that is what we're going to use to try and understand how important these young of the year alewives may be to groundfish populations on the coast. And so that's in a nutshell, that's what we're going to do. And we're going to be talking more about carbon, but we can also use nitrogen as well, even though we're not doing it for this particular study. So next slide. And so this is actually a relatively routine analysis and often in the sense that we're simply interested in what we call bulk analyses of carbon and nitrogen. We literally just digest a piece of muscle and we can get that data relatively routinely. That's not the most powerful way you can do it. And so we do it in a slightly different way. Instead of taking that bulk muscle, we actually separate individual compounds within that muscle tissue. And in fact, these are a whole bunch of amino acids. So this is, we have what's called a gas chromatograph system. And what the gas chromatograph does, it separates out all these different amino acids. And so we can see them here. And then we take those amino acids, and this is separated in time, right? We then introduce the signal into our mass spec. And what that does is it allows us to measure the carbon isotope composition of each of these different compounds as they come off the gas chromatograph. So we're actually now not measuring a bulk analysis, we're actually measuring the carbon isotope values of these individual amino acids. And that gives us a little bit more power in some of the questions that we're asking. And then the other thing that's really amazing is the precision with which we can make these estimates now. So if you imagine that you had 1,000 tennis balls in a bag, and you had 999 of those tennis balls were carbon-12, and one was carbon-13, right? And we measured that. If we then simply add one, we replace one more tennis ball with a carbon-13 and remove a carbon-12. So we're only changing it 1 in 1,000. We can easily detect, would be easy to be able to detect that with the mass spec. So it's a very precise measurement that is due to some really clever people who make these mass specs. Okay, next slide. And so what we're doing is trying then to understand this. And so we're getting data, as Robin said, from the Sentinel Survey. So you've seen that. So let's just go to the next slide and see the important thing for us, right, is that what we're particularly interested in is not so much whether alewives, but sort of writ large are playing an important role in these coastal food webs. We're really interested in whether the young of the year alewives returning, or leaving to the ocean, how important those guys are. And that's a little bit different, right? Because what we're looking at is that great graph, that great image that Pat put up. When we remove these dams, we're opening up a whole lot of freshwater habitat to places where the fish couldn't go. And there's a lot of productivity that happens in that freshwater habitat. We're actually analyzing these carbon isotopes to figure out how much of that freshwater carbon is potentially getting into these food webs. So we're really only, our question can be asked only about how important are the young of the year alewives as they're out migrating from these rivers. And why that's interesting to us, obviously, because of this, this huge amount of freshwater habitat that is getting reopened to alewives by these dam removals. And how important is that connectivity between freshwater and the coastal ocean in terms of sustaining groundfish populations? So it's a slightly different question than, are alewives important? Because our technique wouldn't pick up adult returning alewives being eaten by cod. That would be, that's a different question that our approach is not going to answer. We're really interested in this question of this freshwater production and how important it is. And so this is the first preliminary data of it's a multivariate analysis of the carbon 13 values from a bunch of different groundfish species and for river herring. And the only point really to make here is that you can see that the river herring and the white, so these are again a young of the year river herring, and the green, sorry, they are isotopically completely different from our groundfish. And so what that means is that if these young of the year river herring are going to starting to contribute to groundfish, what we're going to see is that we're going to see our groundfish isotopic signatures start to converge on the river herring signatures. And that is going to be very definitive results that that these young of the year river herring are actually contributing to groundfish populations. A secondary interesting thing for us from this graph was that, as you can see, individual groundfish species actually pretty much cluster together, and they separate out from other species. So obviously on the left-hand side in the cyan color, you've got haddock. Now, isotopically, they're quite different from cod, for instance, which are different again from pollock. So what that means is that these different species are eating quite different things, and that's been reflected in this isotope value. So that's actually got us thinking more broadly about Gulf of Maine food webs and about how these different species are actually interacting within that food web, and how specific the diets are for each of these different species. And so that's another question that I think will be answering from these Sentinel surveys as well. So we're really interested now, I think, in developing a much better understanding of the connections among these different coastal species and where they fit into a more general Gulf of Maine food web. But again, ultimately the question of young of the year river herring, we are now really confident that we've got a technique that is going to answer that, is able to answer that question. We're as you can see from here, we're with Robin, all right? We don't actually, these are our first year samples from 2018. We don't see that yet. We're not seeing freshwater carbon getting incorporated into these food webs. And so the question is, we've got a really good baseline now. And so as these alewife populations start to get bigger, we're in a really good position to be able to start detecting that freshwater carbon getting into those food webs, if in fact it does so. So I think I'm kind of hopeful that we do. I'm also a little bit hesitant, right? Because as Ted will tell you, right, that these small populations that have been fished out, that they kind of had their own culture. And so it may be that the large females that would have been picking off these young of the year herring, river herring, are simply not there anymore. And so it's going to be super interesting to find out, to answer this particular question. And yeah, I'm hoping that there's enough memory within these populations, even given their small sizes now, that they are still able to tap into this resource as this resource grows. So happy to leave it there, but I'm sure we can answer some questions a little later.
Awesome. I'm going to run downstairs so we can get going on this dissection. Please keep asking questions in the Q&A. We want to try to make sure that we get to everybody, but we really want to make sure that we get to the dissection. So keep asking questions and we will get to as many of them as we can at the end. And I'll see you in a second.
All right. Tate, do you have us here in the shop?
All set, yep.
All right. So here's our Atlantic cod. This fish is about 22, 23 inches long. This particular fish came in right at the end of January. One of the things that we've done with the survey is expand the survey into sort of the shoulder seasons and into the winter to try to capture some of these bigger fish. Part of the theory and part of the reason why we're fishing into the winter is that we think that some of these older fish are coming into spawn over the winter, and we think that that's true based on some of the information that we've gotten from the fishermen that participate in the survey and also other fishermen that we've talked with in the community. And so Robin is going to dissect this fish, and I think she's just getting her stuff ready. She's going to grab a pencil so that we can make sure we record all of this information. I'll just keep talking about the fish's anatomy here. Gattids, you can usually tell a gaddid species because it has three dorsal fins here. The codfish will have three dorsal fins. It's got sort of a rounded caudal fin, and then it's got this white linear line running down the fish. So this is an ugly looking fish because it's been in the freezer. Usually they're a lot prettier than this when they come over the side of the boat. But okay, go ahead, Robin, and let's show everybody exactly what happens on board.
So first thing that we need to do, just with the Sentinel survey in general, is take a length, weight. So I'm gonna put this nose all the way to the edge. See right here. And I'm going to call that 61, even though it's a little bit less than 61. Essentially, if it's over the 60 line, we round up. So 61 centimeters. I'm writing all of this down. The organization key. We want to make sure that we can keep all of these samples straight with the fish that it comes from. This guy here. This guy was just about 6 pounds. So first we want to find out whether it's a male or female. Do that. Coming into the fish just here. Fair warning, cutting open a fish can be a little bit gross. So if you are a little squeamish, just be prepared. So this is a female, and I know this because all of this right here, those are eggs in there. So while this female is not currently spawning, if she was spawning, I could push on this and we would see eggs coming out. But this is what we would call developing or pre-spawning. So she's mature, but she's not currently getting her eggs out there. And you see that it's filling up the majority of this cavity. When fish like this are spawning, I mean, it can fill up an even bigger area. So I'm just gonna write that down. This is a
I forgot to get you a pencil.
It's okay. Developing, which I'll write down in just a moment. So we have that. The next thing that we're going to do, I would normally be taking a picture, but we've got lots of pictures happening at the moment. And I'm actually going to switch knives, I think. And it's a little frozen, so we'll see how the cutting into its body goes. But so to take a sample, the stable isotope samples for Simon, we're gonna just, here's the dorsal fin here. Just gonna cut a little bit ahead of that into the fleshy part. This is definitely a bit too big of a piece, so I'm gonna cut this down a little bit, like that. File here that we're gonna put it into. And of course, I'm not going to write up a million labels right now because that's kind of the most boring part of this whole thing. But we put a label with every single one of the samples, and we know exactly what fish it goes to. That. Next, we're going to do otoliths. I am not a pro otolith taker. We'll see if I can even cut into this guy's head. But otoliths are fish ear bones. And essentially, it's how we age a fish. So, this guy. I'm usually just cutting forward on his head here because the otoliths are in the brain cavity, which are up here. Almost there. So we've got the brain right here. And the otoliths are just kind of on the side here. I've got to cut a little deeper. We've got a strong head to be able to take a look in here.
Oh, tweezers.
Tweezers, yeah.
I'm gonna grab Robin some tweezers real quick. I don't know where they went. I'll swap. Don't worry about your hands.
That's it. All right. Pat's going to provide the muscle here for a moment. So we get into here. So this is an otolith. And otoliths are found in in all bony fish species, so not in things like sharks. But they're really cool because when you cut into them, they have these rings, like rings on a tree. And by counting the rings, you can age the fish, which is, that's always like my favorite fact because it sounds like a joke. But so these are really important because we can link things like age to length and age to weight and age to maturity, all of these other things that we've been looking at, which are really, really important things to know because this is the only way to age a fish. So I will take these, we'll swap it on up.
So fit. That's a big one.
So next thing we're going to do, thin clips. So I mentioned these are a way that we get DNA samples. And so there's some really incredible work going on right now with looking at cod DNA and what that means, because basically what we can get from this information is where these fish are born, where they come from, which is really essential when we're trying to define things like fish stocks, which basically just means because like we mentioned, we haven't really seen spawning cod in the Eastern Gulf of Maine for a long time. So like, where are these fish coming from? Are they coming from Canada? Are they coming from the Western Gulf of Maine? Do we have our own spawning stock? Looking at differences in DNA, they can kind of start to piece together that information. But now for what we're all here for, which is taking a look at the stomach. Looks like there's something in there. So this is the liver right here. Which, yep, this whole big fatty thing right here is the liver. And this is the stomach. So you'll see the stomach leads all the way to the throat, of course. And this thing is called, that looks like a whole bunch of little noodles or rice or whatever you want to call it. It's called the pyloric caeca, which helps actually kind of crush up a lot of the stuff in the stomach before it goes to the intestines. So I'm going to cut out this stomach. Take a look at what we've got in here. That green thing, that's spleen. All right, so we're going to cut off the pyloric caeca, and then we're going to see what we got in here. It's funny for this big fish that it looks like it has such a small stomach, right? Because, and it, I mean, these things really stretch depending on how full their stomachs are. Like I've seen tiny little cod with absolutely huge stomachs. Apologies, this is a little frozen, so I'm trying to get this guy open a little bit more. What we got in here? A whole lot of little bits. Here. Hello, welcome to our What Are Cod Eating 101. We certainly have, I mean, we have some animal remains in here. It's kind of hard to tell. Like I mentioned, sometimes there is a lot of things that are decomposed, but what's interesting, this right here, this is an otolith. So this fish ate another fish. Because that right there is an ear bone that we were just looking at, but that came from a much smaller fish. And so sometimes, like obviously if you're looking at this stomach, right? I can't tell just by sight what this came from, but we can analyze otoliths. I could send this in
and we could find out what kind of fish that was that it had been eating. Because of course, these hard parts are generally what stick around longer, you know, than all the soft, squishy parts. Let's see if I can find any. There's some little shrimpies in here somewhere. Now it looks like this guy was pretty hungry when he was caught. So otherwise we don't see a ton in there. But that's, I mean, seeing that otolith is a really, you know, is proof that this is a fish that is eating other fish, which if you guys remember from a lot of my analysis, we're usually finding that fish about this size, maybe a little bit smaller, but usually not fish much smaller than this are eating fish. So I'm gonna put, just so that I can kind of pick through this later and make sure I didn't miss any little pieces in here, I'm going to put the stomach in ethanol jar along with that otolith because I don't want to forget that. And there you have it. Those are samples that we take.
Cool. So Robin and I are going to head back upstairs now and sit back down at the laptop, and we can take some questions.
All right, so we got a question from Randall Curtis. Randall, are you ready to talk? I'm gonna get you on here.
Unmute. Yeah, I'm.
Hear you. Perfect.
So I had a couple of questions. One was I was told that diadromous trout scales in Alaska. I was involved in a creel survey a number of years back. Could also tell age. Is that true? You mentioned it was just really the otoliths, but we were collecting a lot of fish scales during the week that I was involved in it, and that I was told, if I'm remembering correctly, that fish scales, at least in this species of trout, could also tell age.
That could definitely be true. Yeah, especially in a species like that has like kind of very defined movements year to year. It's definitely possible that I don't know whether it's like the size of the scales or maybe, you know, that moving into those different environments kind of changes the compounds. I'm not exactly sure, but I would not be surprised to learn that there are at least some species that you can tell from the scales. That's really interesting. Thank you.
Yeah, thank you. That was fascinating. That was a good job.
The fish was still pretty frozen too, so it was kind of difficult to get down through it. But usually they're a little easier than that.
Hey, Robin, you should tell folks that you can actually, you know, that otolith, the otoliths are this distinctive, right, among the different species. So you can not only tell that it's an otolith, you can actually tell which species of fish that otolith came from, right, for your gut content study.
Yeah, and even sometimes within species, like I know there's been some research, I forget if it's on cod, but just cod living in different areas, that their otoliths can look a little bit different, which I just think is incredible. And also that you would kind of expect that the bigger the fish, the bigger the otolith, but tuna have these teeny tiny little otoliths. They're very hard to find. And they're huge heads.
In a big
Very big.
Yeah.
I found my second question. It was about the difference between jigging and long line, and what do fishermen prefer in what types of circumstances?
Well, it depends on who you talk to, and it also depends on the age of the person that you're talking to. The gear that we use on the survey is rod and reel gear for jigs. Basically, the jigs are, it's a 26-ounce Norwegian diamond jig. It sits about this tall. I should have grabbed one while we were down in the shop. I've got a pile of them down there. But it's got a couple of different teasers and flies on it that sort of dance around in the water. And the long line is a 200 hook series of baited hooks that sit right on the bottom and anchored off on both ends. The commercial fishery is typically executed with either drags or gill nets. And all of the fishermen who used to fish out of Stonington commercially, almost all of them fished with gill nets. There were a few draggers here and east of here, but primarily it was a gill net fishery here in Stonington. A lot of the recreational fishery will execute that fishery with mainly jigs or long lines, and then the commercial fishery uses drags and gill nets.
Yeah,
Great.
I will say it's fairly uncommon for us to catch cod on the long line. It could just be a matter of the size of the hooks or the type of bait, but we almost always catch, or it could just be that generally these long lines are operating in deeper water, and we know that cod prefer shallow, but cod are also, visual hunters. So the jigs have this like big, like silvery weight on the bottom that they can both hear and see bouncing off the bottom. So if they're really interested in catching cod, then they'll probably prefer jigging, but there is certainly a much greater capacity to catch more fish, and to catch a more diversity of fish on the long line. So it depends.
Thanks, Randy.
Let me know if you ever have a party boat going out with tourists or amateur scientists. Love to go.
Awesome.
Sounds great.
Thank you, Randall. And just to add to that, Josh Dime, who's one of the fishermen on the Sentinel Survey, put in the chat, the long line is way more fun, at least when you catch something. So.
Hey, Josh.
It can be a little hit or miss.
So that's a great opportunity to ask a fisherman a question. If anybody has any questions for Josh, he's on too. So.
Great. So we have a question from Peter about what does the circle in the center represent on one of the last slides. So Pat, I don't know if you could pull up the last slide on Simon's presentation while I get to another question. We got a question from David. Are the young of the year alewife and herring being eaten by stripers, bluefish, or Atlantic salmon? Simon or Robin, you want to take a stab at that?
Could you repeat that one more time?
Yeah, absolutely. Are the young of the year alewife, herring being eaten by stripers, bluefish, or Atlantic salmon?
So me and Simon actually both recently attended essentially a conference, trying to combine all of the information that we know from people studying diet. And we've had kind of a hard time identifying young of the year river herring in pretty much any of the kind of main predators that we expected. I don't remember specifically about those species. Simon, I don't know if you do either.
I didn't. Yeah, I remember the same way, Robin. That there's not a whole lot of data in terms of the young of the years. Although you can imagine stripers would be caning those little we call them peanut bunker down here, right? I mean, stripers love those guys. So they're
yeah, especially when the young of the year are leaving in the fall. I mean, they leave in the millions, and it's usually after a significant rainfall event when the ponds sort of fill up to the level where there's enough water in the stream to carry those young down to the ocean, and they pretty much leave all at once. So you can imagine just millions and millions of little baby alewives leaving. There's going to be some stripers out there with hungry mouths open. Simon, there was a question about the circle in the middle of your plot here.
Yeah, so sorry, this is I didn't want to go into the details, but this basically just is simply a way of representing what's actually a five-dimensional five-dimensional data set in 2 dimensions. So the small dots at the end of the lines, they're all individual fish, right? So we have 5 haddock, and we have 5 cod and 5 white hake, and then so if they're close together in space on this diagram, that means that they have isotope signatures that are very similar. And if they're further apart, then the isotope signatures are different. So that's the circle in the middle actually doesn't represent anything necessarily. This is simply showing, again, a 5-dimensional data set in 2 dimensions so that we can just see it. But the point is that those small dots that are close together indicate that those fish are eating very, very similar things, and points that are a long way apart are eating things that are very different. And so again, the take home from this for then was that haddock, individuals, haddock are doing very similar things, one haddock to the next haddock, but that's actually very different than cod in the same location. So there's some really interesting questions to get at with that.
We are right at 1:30, but if folks have more questions, we've still got 35 participants online. So for whoever wants to stay and ask questions, we can stay on for another 10 minutes or so and answer those questions. So go ahead with the next one, Tate.
Great, thanks, Pat. So this is from Bob Senek. Hi, Simon. In terms of trying to understand why cod stocks have not increased despite the absence of fishing, seems to relate to deficiencies in their diet. Do we have any evidence that cod are food limited? Might populations be predator limited, e.g. from seals?
Stanek, are you still on? That's like a total ringer question. I can't believe you did that to me.
Yes, yes.
So you know what? I'm not sure why cod populations have not recovered, Bob. You this better than me, right? It could be from seal predation. I'm not sure that I necessarily buy that. It certainly could be, they could be food limited, or perhaps the populations might recover quickly if they had more food. Maybe that's a better way of putting it. We're not really necessarily trying to address that specific question. I think we're more interested in the question of whether the cod populations increase or not, or the groundfish populations in general. Can we simply pick up that freshwater productivity that is now moving into the coastal ocean in the fall with those migrating young of the year fish? And so I'm sure we would, I would hope that increased food supply might help cod recover, but no, I exactly why populations have been, are still subdued. That's a question. That's a bigger question, and goes to perhaps some of Ted's ideas too, right? In terms of the loss of effectively the culture of those small populations, and that if that's the case, then wow, it's hard to know when they might recover, because that's going to take a lot to come back from. Yeah. But yeah, very good question, of course, Danik. Thanks.
I will say I can't really speak to predators, but a decent part of my PhD is going to be kind of focused on looking at cod diet changes through time, and whether we can kind of relate this to differences in condition factor, whether we're seeing kind of smaller weights at the same length or smaller sizes at the same age or maturity, and a lot of that is actually based off of your work, Bob, on ecosystem shifts towards kind of more of a invertebrate dominated as opposed to kind of these more fish dominated areas that cod used to be. So stay tuned.
Great, thank you,
Bob. Thanks for the heart attack analogy. By the way, I use it often. It's perfect.
We got another question from John Anderson. Is plastic or other man-made waste found in cod stomachs? What and any trends?
So of the 93 stomachs that I've gone through, one that I analyzed last February had a lobster band in its stomach, which pretty much took up the entire stomach of this particular cod. But I will say, generally, we're not finding lots of plastic in cod stomachs, at least that I've identified, beyond lobster bands. Whether that's because they're just passing it generally. I think since they're bottom feeders, I think it's likely that they're at least picking them up. But as far as I know, I don't think that there have been any studies kind of quantifying that or trying to understand whether that's, impacting things like growth or, reproduction, some of these other things that we know that plastics and pollutants can cause. So personally, I have seen evidence, but it's not a terribly common thing. But also remember that these stomach analyses are really a snapshot. I mean, it's what they ate essentially that day. So it's kind of a very minimal view, whereas Simon's work is kind of a much broader view of generally what they've been eating over a longer period of time. So.
Speaking of which, we had 4 different codfish that ate that had rocks in their stomach, and I had heard from a fisherman a couple of years ago, and I thought he was crazy, but he said that codfish and halibut and other large sort of bottom feeding creatures like that will actually eat rocks to keep themselves ballasted on the bottom in anticipation of a big storm or a blow event. And I thought he was nuts until I saw the pie graph from Robin this spring. And I went back and looked at our data and cross-referenced it to the weather at the time. And coincidentally, there were only 4 samples, but they were within 2 days of the wind kicking up to about 25 knots. So I thought that was pretty interesting that they had rocks in the stomach within a couple of days of a big blow event. Definitely need some more data on that, but I thought it was pretty interesting.
That is super interesting. We got a number of questions, so we'll keep going. This is from Robert Ferrero. Do the cod migrate, and if so, how far and to what temperatures?
So as far as the Eastern Gulf of Maine, since we don't really see very many spawners anymore, I would say that seasonally we probably aren't seeing as many shifts. I mean, generally as cod get larger, we find them over deeper habitats, and that, you know, historically during spawning seasons, which I think are generally in the spring, I'm trying to remember when the local populations around here were spawning, that they would move in. But like I mentioned, it's entirely possible that almost all the cod that we have in this area came from somewhere else. And we haven't really seen large cod in this area for a long time. So I think it's very likely that there's movement in and out of these areas, although I think kind of in their recent low abundances, that's kind of less understood than it has been in the past.
Yeah, it's part of the reason for the genetic studies that we do. We can sort of zero in on different subpopulations using genetics, and it's also the reason for the photographs that we take. So Robin mentioned that she lays the fish flat on a measuring board and takes a photograph. From that photograph, our partners at the Gulf of Maine Research Institute will take about 19 different measurements of the fish to get an idea of the body morphometrics. And we can tell if that fish was a migratory fish or a sedentary fish just based on the body shape. Good question, thank you.
Great, thank you. This is from Libby Jewett. Is there any opportunity for citizen scientists to get involved?
Hey, Libby. Yes, there is. Actually, there's a lot of opportunity for citizen scientists to get involved. Thanks for the softball. My colleague, Mike Thalhauser, actually does alewife counts on the streams right here in the Bagaduce River. So if you're local to the area and you're near the Penobscot River or the Bagaduce River, he's always looking for volunteers to go count alewives during the alewife run in the spring. So that's one way. And we also, well, that's probably the best way. And then obviously take down our information on the last, did we show the slide? Well, we can send it out to people, but get in contact with us if you're interested in hearing more, and I'm sure we can find some volunteer opportunities for you. I could use some help baiting hooks through the summer. So if you're interested in volunteering to bait hooks, I always use help in the shop. So thanks.
Great. And then we got another couple questions. One from Josh. What is our confidence level on when and where the cod are spawning?
As far as the Eastern Gulf of Maine, our confidence level, I would say, doesn't really exist, because we quite honestly have not had hard evidence of spawning cod in this area for several decades. You know, we hear fishermen talk about it. You know, we do see cod that we think are probably the right size to be spawning, yet we just aren't really seeing it. However, there are several spawning areas in the Western Gulf of Maine that we have pretty well defined at this point in terms of both genetics and kind of other things there. But Eastern Gulf of Maine is a huge just question mark as far as spawners go. It's, I think you were mentioning, so a recent working group for defining cod stocks in the Gulf of Maine recently suggested that the Eastern Maine be considered separate from the Western Gulf of Maine. And part of that was because we essentially don't have a spawning population, at least that we know of. And that does make us a very different area with a different, you know, kind of diversity of species and cod movements and cod ecosystem dynamics than the Western Gulf of Maine does. And that, you know, if we're just kind of assuming that everything is happening the same, we're probably missing some pretty important information that's unique to cod in the Eastern area. So
yeah, at the same time, you know, even though we don't catch very many adult-sized fish, we do catch a lot of juveniles, and those juveniles had to have come from somewhere. So whether the adults are moving in and spawning and then leaving, or there's, you know, there are some adult population of fish that we just haven't pinpointed yet. We just don't know. The current thinking is that the adults are moving in from another place, spawning, and then they're either leaving because they don't have enough feed to keep them happy, or another reason, you know, they might be migrating back to the places where they tend to like to hang out.
yeah, that is the question.
And last question, another one from Bob. This is about diet species identification. When you get a mass of poorly defined biomass, would eDNA barcodes help identify what they had for dinner?
So we've talked about the possibility of using eDNA. As far as I've kind of become aware, the confidence of eDNA, at least in an ocean setting, is kind of still kind of questionable, because it seems like a lot of that has been tested in freshwater settings. So it has certainly been talked about, but you know, there are obviously concerns about, you know, how long are those DNA markers staying around? Has it been a long time, or are they just sweeping out right away and we can't? I mean, we've even just talked about using eDNA to try to validate, you know, what we're catching on the survey and seeing whether there were other species down there and we just missed them. So the idea has certainly been floated around. As of now, there aren't hard plans to include that, but definitely, you know, a really interesting part of research that I think would be really helpful, if not now, then in the future.
Yeah, we've actually just done some you know, it's really meta barcoding of gut contents rather than eDNA, right? And we're not sampling the water, we're actually sampling DNA in the guts, and it definitely works for sure in marine environments. The problems are it's still presence absence, right? It's very hard to get a quantitative signal out of gut contents. So we can say the DNA is present. It's really hard to say, you know, how much of a particular prey type was present in the gut. It's possible to get around that with some quantitative PCR techniques, but it's still a little bit tricky. But it's definitely there's a lot of interest in it, and I think we'll see more of that in the future for sure.
All right, and with that, I think we're going to conclude. Thanks, everybody. Round of applause for our panelists, Robin and Simon, and thanks, Tate, for running tech for us. If you want to learn more, come visit us at coastalfisheries.org. Get in touch with our executive director, Paul, or myself, and hit follow on our social media pages, and definitely come to future Lunch and Learns. Thanks for coming.
Thanks, guys.